Dear all, wherever you are, I hope you are safe and well in these very particular circumstances and challenging times. My name is Karim Benham. I am Associate Professor in the Center of Biotechnology at the Technopark of Borj-Cédria in Tunisia. I am the coordinator of the PRIMA project entitled Development and Optimization of Halophyte-based Farming Systems in Salt-Affected Mediterranean Soils, in short, HaloFarMs. This project is coordinated by the CBBC, the Center of Biotechnology in Borj-Cédria, in collaboration with the Tunisian Center of Water Researches and Technologies, the Egyptian Desert Research Center, the Spanish Research Council, the University of Occidental Brittany, and the National Institute of Research for Agriculture, Food, and Environment from France, the University of Pisa from Italy, and the Center of Marine Sciences from Portugal. To celebrate the kickoff of HaloFarMs, we are pleased to organize today this virtual meeting, hosted and facilitated by the National Agency of the Promotion of Scientific Research, ANPR. After the opening remarks, we will have a general presentation of the PRIMA program and the role of project monitoring by the PRIMA project officer, Dr. Fabrice Den-Trestango, followed by the description of HaloFarMs by the coordinator and the partners. Before we start, we would like to already inform you about few general rules that apply during the webinar. Cameras and mics will be deactivated, except for facilitator and speakers. You will have the possibility to ask your question in the chat box. The webinar will be recorded and will be available in replay so that you can watch it at your convenience. Now, to open this meeting, I am honored and pleased to host first Professor Chadli Abdelli, the General Director of the National Agency of the Promotion of the Scientific Research at the Tunisian Ministry of Higher Education and Scientific Research. Professor Sharfi, which also should come to open this meeting, just apologize and she will not be able to join us. So I will give the floor to Professor Chadli Abdelli to open this meeting. Please, Professor Chadli, the floor is yours.
Thank you very much, Karim. Dear colleagues, dear friends, it is with a great pleasure and honor to be with you in the kickoff meeting of our project, HaloFarMs. I am particularly happy for the following reasons. Firstly, this project is about halophytes, and you know that personally, I started working on this plant almost 40 years ago, first with a small local team and then in several international networks. I particularly mention the project of Professor Halophyte-based within the framework of the concerted action Sustainable Halophyte Utilization in the Mediterranean and subtropical dry region, launched in 1996. Some of partners of HaloFarMs were involved in this action, like the team of my colleague, friend, and brother, Hassan Shah. The results obtained on halophyte in this project and other studies demonstrate their diverse economic and ecological interests and justify the growing interest given to these plants as promising candidates for creating new productive area in marginal zones. Our project, HaloFarMs, fits particularly and perfectly into this approach. I am also happy because the funds allocated by our Ministry of Higher Education and Scientific Research will be managed by our National Agency for Scientific Research Promotion, NPR, that I currently manage. This agency constitutes a national interface structure aiming to bring research closer to the socio-economic environment and to put research at the service of societal challenge. Our project, HaloFarMs, is a good example of transfer technology from laboratory to socio-economical partners. In fact, the objective of our project HaloFarms are to promote the valorization of research results and the know-how on halophytic species. Another reason of my great satisfaction is that HaloFarms project has results in the constitution of a very good, very strong consortium involving friends and colleagues from several countries: France, Italy, Portugal, Egypt, Spain, and of course Tunisia. At the local level, I am very happy that the laboratory of extremophile plants that I founded in the Center of Biotechnology Borj-Cédria is the main coordinator of this project. I am very proud that my scientific descendants in this center, represented today by Dr. Karim Ben Hamid, General Coordinator of this project, and Dr. Ahmed Dabbez, Director of the Laboratory of Extremophile Plants, continue and diversify their approach to progress in the characterization and valorization of these plants. This laboratory is considered and recognized today as a structure of excellence in this field. Congratulations. Full success for our project, and thank you so much for this kind invitation. Thank you.
Thank you very much, my Professor Shadi Abdelli, for these opening remarks, and we hope that our project will be successful. Now, I will give the floor to Dr. Fabrice Denstringel from PRIMA. He is the PRIMA Project Officer, Farming System. He will give us his opening remarks. Please, Fabrice, the floor is yours.
Thank you very much, Karim. Thank you, and good morning or good afternoon to all of you. I'm very pleased to be here. So I thank Karim for the invitation. It's an important point in your project. The kickoff meeting is very important. It's the occasion to meet, even if obviously this year it's a little bit particular and we have to meet online, but congratulations to organize this kickoff. And I also want to thank Shadli Abdelli, for the facilitation of the organization, and we know it's also an important partner for PRIMA in managing the fund of the Tunisian project. So what I want to say just to start, it's congratulations, because your project is a very nice project. Project on halophyte, I think, are really on time. It's a way to valorize maybe resources that were not looked at. Also a way maybe to have the possibility to use new fields that are affected by salinity in the Mediterranean. We know that this problem is appearing everywhere. So really congratulations for that. Just to give you some numbers, there were 160 pre-proposals submitted. Only 30 have been funded. So you are part of this 30 project, and you are now also part of the group of the PRIMA project. We have now 83 projects running for a total budget of more than 100 million euros that have been injected in the region. So we are very happy, proud of this success. And I wish you a very successful project that is, as I said, timely. And I also have a personal interest because I'm an ecologist by training. So, of course, this kind of study, I like it personally. So, thank you. And I give you again the floor, Karim. After I will intervene to present you the monitoring of the project, as I am the project officer in charge of follow up your project. Thank you very much.
Okay, thank you, Fabrice, for these kind words and for these opening remarks. Now I will give the floor to my colleague, the director of the Laboratory of Extremophile Plant in the Center of Biotechnology of Borj-Cédria, Dr. Ahmed Dabbez. He will give as his opening remark, but also he will give us also an idea, general idea about the Laboratory of Extremophile Plant in CBBC. Please, Ahmed, the floor is yours.
Thank you very much, Karim. First of all, I would like also to warmly welcome our participants and also our partners, or even friends who are involved in this nice and big project. Actually, as mentioned by the Dr. d'Entressangle, I mean, the kickoff meeting is very important in this project, and it was supposed that we will host this event in Tunisia, and we were really happy, and we were expecting, we were looking forward to meeting you and to discussing here directly and meeting together in Tunisia. But well, because of these circumstances that we are, prevailing since 1 year, so we are obliged to adapt, and I would say, and that's why also we are at least we are happy that we can meet using the webinar, and we are in this way. I'm really thankful to Professor Abdelli from the UNIPI for the facilitations and for all the support that he gave us in order to be able to help this event. Well, as Karim said, this project is quite important. I mean, from different dimensions for at the international level. I mean, with respect to the Mediterranean area, but also to the our laboratory, which is, I mean, for the first time, it's that we are coordinating such a big project. We were already involved in several international projects, but it's really the first time that we directly manage and coordinate such a big project with almost, I think, 6 or 7 partners from different countries. And this is well, this is a challenge for us, but I'm really confident that Karim and with our support, we will succeed in coordinating this project. The second important dimension is that this project will really, it's an asset for us. It will really enable us to further extend our international collaboration with several partners. Some of them, I mean, we already know since we have been involved in for several years in joint projects. But this is also a nice opportunity to further strengthen our collaboration and to expand our network, especially in regard to halophyte valorization, which is, I think, up-to-date topics. I mean, especially in the context of the climate change, that we are already seeing the impact of these changes, especially at the Mediterranean level. And also, with respect to our laboratory, I mean, so the LP is known for more than 20 years for being the first and so far the only research structure in Tunisia focusing on the characterization and the valorization of extremophile plants, namely halophytes and xerophytes. And during all this period, with the strong support of Professor Abdelli, who founded this laboratory, so we gained, I think, well internationally recognized expertise in this field, and we gained many data about halophytes. I mean, either at the fundamental level, and we gained many results, and I think we contributed to the better understanding of halophyte adaptations in their environments. but also at the applied level, I think we also contributed in the way of really showing that the potentials of these plants, of these precious resources, is highly and could also concretely be valorized at the economical and environmental level. So we are, for instance, in our lab, so we have 4 main projects. We have one project which is aiming at valorizing halophytes as a source of fodder. We have also another project on halophytes and their role in the phytoremediation against, for instance, heavy metals. And we have a third project on the halophytes as a source of foods, and the final project on halophytes and their on the understanding, on better understanding of how halophytes can adapt to their natural conditions. I mean, and this is rather at fundamental level. So a major keyword for us was always besides the characterization, was also the valorization of halophytes. And I think this is, as far as I know, one of the rare opportunities for us that will enable us to work with local farmers here in Tunisia. And this is really, our project is really pioneer in this way, since it will be the first time that we will work with local farmers in order to concretely show that halophytes can improve soil structure, but also can contribute to desaline water, and then to improve and also contribute to creating maybe new farming systems in marginal areas. And I'm really so there from my side, and also there are from other sides, there are also many expectations in this regard. And I'm really convinced that we will succeed in these tasks with your support. I mean, with your expertise also. And this is for us a really strategic or a strong milestone in our strategy, consisting at transferring results obtained at the laboratory to larger scale, and to create, as I said, win-win partnership with local farmers. So this is this was my general words, I would say. But once again, I would like to really welcome all of you, and I hope that we will be able as soon as possible to meet directly in Tunisia. This will be a great privilege for us to meet you, and I wish you all success in this how to say adventure, I would say. But I'm really once again convinced that we will succeed in this. And so thank you, Karim.
Thank you very much, Ahmed, for these words, for this overview of what we are doing in the laboratory of plant extremophile. Our laboratory is one of the famous laboratories and the first laboratory working on extremophile plants. And I thank you all. I thank all the guests, the invited guests, for their opening remark. Now, you know, HaloFarms is a part of PRIMA program. It is centrally organized by the PRIMA Foundation. Though it is very important to know what is the PRIMA program, what are the general rules to monitor project in the scope of PRIMA calls. So I will turn it again now to Fabrice, the project officer, PRIMA project officer, to give us a presentation about PRIMA program and the rules for project monitoring. Again, please, Fabrice, the floor is yours. You can share your screen, please.
Thank you very much. So I will share my screen to show you the PowerPoint. Okay. Could you see the screen?
Yes, it's okay.
Yeah. Okay. So I will start because, yes, as reminded, you are part of the PRIMA program. So PRIMA will monitor your project. And there is just a small point I didn't mention in previously, but also I'm very pleased to see this project that is coordinated by Tunisia, because in PRIMA we of course want to have an action on the southern countries, and what we are seeing there is there is few coordination of the project by a southern country. So for me, this is also a very important and good point. So I wanted to highlight this before starting. So now I will present you how the monitoring of your project will work. This is more or less the outline of the presentation. So how the reporting will work, some words about the financial aspect, and some words about the dissemination of the result and the open access of your data, and how to acknowledge PRIMA and your funding agency in your communications. So to start, you are in section 2, and all the project of PRIMA will be monitored through a platform, a monitoring platform that is called the MEL, Monitoring and Evaluation and Learning Platform. This platform has been developed in partnership with ICARDA, because in reality we wanted to try also to promote your project. So it's an indirect manner of promoting your project, because you will be integrated in the ICARDA database. I mean, if somebody is looking for research dealing with halophyte, for example, by looking inside the ICARDA database, they will find also your project. So we thought it was a good idea to have this kind of synergy to valorize the database and maybe facilitating the collaboration. So to go back to practical aspect, the coordinators and the partner will have this access. It's a platform, it's online, so it will be always open, and each one will have an access with your email and a password. How to do that? To all the coordinator, we will send an Excel file. This Excel file, in reality, we ask you to be able to inject automatically all the data of your proposal inside the platform, on the platform, to avoid you or the coordinator especially, to enter like name by name of each partner, to enter each work package, each deliverable, etc. Instead of doing one action by one action on the platform, we ask you to fulfill this Excel file. So this Excel file has different tabs, as you can see, general information. So this is just the name and the starting date of your project, more or less. I'm simplifying. After, there is a tab on the partner, so the list of the partner, the work packages, the list of deliverable, the list of tasks, the milestone, etc. In reality, these tables are already in your proposal. We try to make it as simple as possible. So normally you just have to copy paste all the information that is already inside your proposal in all the tables that are included in your proposal. So once we will receive that at PRIMA, we will be able to inject all this data directly in the platform. And when you, the coordinator or the partner, will log, you will be able to see all this information. Of course, sometimes there are some small mistake you want to correct, or you have to add something. There will be this possibility. The coordinator will be able to edit the field directly in the platform. There is different section, like the general information about your project, so the acronym, the summary, the duration, these kind of things, the budget. And after, there are several parts more specific to your project. So the objective and your target, the location of your study site, or if you have demo site, all these kind of things, pilot, etc. You can put this information there. There is a space also to upload all type of media you can generate during the life of your project. It can be posted on Facebook, Twitter, all these kind of things, but also video, TV, interviews, etc. So you will have this opportunity to upload all your files there, key documents you want to share with your partnership. The strategic framework, this is the Gantt. It's the Gantt diagram, more or less, so it will be automatically generated by the platform according to the date that you will have to enter and some part of the note. But just to let you know that everything can be edited by the coordinator. The coordinator is the one who has the right to edit. The partner will have the right to see only, not editing. For the periodic report, PRIMA is expecting two reports. One at midterm, so at month 18, and one at the end of your project. So your project is a project of 3 years. So here I put the date. I believe you started the 1st of October this year. And so the 18 months will be the 1st of April 2022. This is when you will have to submit your midterm report. For this also, you have 60 days to submit it. It means you have from month 18 to month 20 to submit your midterm report. And it works exactly in the same way at the end of your project. At the end of the 3 years, you have again 2 months to send us your report, the financial and technical report. Here, what you can see, there are a yellow bar or orange bar that is called the continuous reporting. So this is very important. As I told you, the platform we will use will be accessible at any time. So it's very important for the coordinator to upload each time he has a new deliverable, to upload it directly on the platform. Because everything that has been already updated on the platform, you will not have to do that at the end. You don't have to wait month 18 to gather all the deliverable and to put everything at the same time, because this is a huge amount of work. So it's much better to use this continuous reporting period. If at month 6 you have a first deliverable, put it online, you upload it, and so you can forget about it. It's done. Your deliverable has been uploaded. So this is very, very important, and I really encourage you to do that. In the platform also, as you will have to enter the date of the deliverables, you will receive a reminder 1 month before the date of the deliverable. The platform will tell you, hey, don't forget you have this deliverable that belongs to this work package to submit in 1 month. So you will be informed. What you cannot do on a continuous basis, it's the midterm report itself. But I will explain you what is this. So for the periodic report, you have the technical and the financial report. I will first start with the technical report. The technical report has 2 parts, part A and part B. And the part A is all the data you will have to fulfill online, meaning it's again the same table you have in your proposal. So I will show you. Are expecting in this part. So first, a summary for publication. I warn you, this summary will be publicly available, so you don't have to put any confidential data here because this will be published online. And as I was telling you at the beginning, we have developed this platform with the ICARDA, and this platform will generate a fact sheet for each project, meaning you will have like a kind of small web page with the name of your project, the coordination, who is the coordinator, the partners involved, and this summary. So this will be like the identity card of your project. So you have to write it carefully and not make a big summary, because if not, people don't read it. But this is why I insist, be careful, it's a public summary. This summary has to be structured in 3 parts. The first part, it's the context and the overall objective of your project. So it's the beginning of your proposal, if I may say. The second part is where you explain all the work you have performed from the beginning until the reporting period. So if it's month 18, it's the first 18 months. At the end of the project, of course, it will cover the full life of the project. And the third part, it's the work you have to do, that is to achieve the impact that you have planned in your proposal. So this is how it will work. And this will be to be filled online. After, as I told you, these are exactly the tables that were in the template and in the proposal. So you already have this table, and when you will log to the platform, more or less, you will have your work packages, the list of deliverable, etc., the name of the work package, the expected date of delivery, etc. And so in front of each line of each deliverable, you will have an upload button to upload the corresponding deliverables. It will work exactly in the same way for the milestones. Here for the deliverable, of course, we understand that sometimes the things don't work exactly in the way you have planned it. So you may have some delay, or you may have to reschedule some part of the proposal. So there will be this option directly in the platform where you will have the column of the the foreseen delivery date, and you will have the option there is another column with the new expected date, so the new rescheduled date. So for the milestone, it will be the same. We will ask you if you have reached your milestone or not. If there are some corresponding documents, you will be able to upload them on the platform. Ethical issue, if there are some ethical issue, also you have to report that, and all the part related with the management of the risk, the mitigation action you can have to develop according to the context. For example, the COVID, I think it has affected many projects, and so it has to be, it can be considered as an unforeseen risk. And of course, if you have an unforeseen risk, you have to propose a contingency plan, so a plan B to be able to reach your initial objectives. And the last part to be fulfilled online is the list of all the not the deliverable, but the scientific papers, the publication, the book, the conference, the training, all these kind of things. So scientific publication and dissemination of the results. This is what you will have to fulfill online. For the part B, so the part B is the report itself that we are expecting at month 18. The part B, here we put around 25 pages. It's not mandatory, but what we want to insist on is don't write a PhD thesis. This is not useful. We want something very practical, and I will show you. So the template will be available on the platform, and it's a kind of classical H2020 template without the table, because the table will have to be fulfilled online. So this is why we want something that is concise, that is short, and that is precise. You have to explain what you have done, so per work packages, how the work has been carried out, and the progress you have done in each work package. You have to update the plan for the exploitation and dissemination of results, as well as the data management plan, because these are things that are evolving according to the life of your project, the timing of your project. So you have to update that. And here in point 4, you can see follow-up recommendation and comments from previous reviews. This will not apply normally for the first report. But how the things will work? At month 18, we will give all your deliverables and your report to an external expert that is expert in the field. And so he will review your project and he will make a small report telling us that if you are online with what you have planned, or if there are major concern or whatever. And so he can make some recommendation and comments. We will, of course, send you this information, and in the next report, you will have to explain if you have taken into account the comments or recommendation, why not, or how you have done it. This is why we have this point 4 years. And after, in this small document, also you have to explain all the deviation you have from the original proposal. It's very, very important, I insist, to have a transparent collaboration. If you have problem, it's not a problem. We have to be aware. We will try to do as much as we can to help you. Our goal in PRIMA is to help you. We want your project to be successful. So I insist on that. If you have any problem, any doubt, anything, let us know, because sometimes we can help you and facilitate the things. If you tell me everything is wonderful, but when you are reporting, we are seeing that nothing has been done, maybe sometimes it's too late to act. So for this reason, I really want to send you this message. Be transparent. If there is a problem, it's not a problem, but it's better to speak about it and try to find solution instead of hiding it, and at the end it's a bomb and we cannot do anything. So this is the message I want to carry on. This is for the technical report. So I finished with the technical report. For the financial report, as you are in section 2, you all have signed national contract with your funding agency. So what I'm presenting here, it's what PRIMA will do. PRIMA will monitor your project at the global level, but it's totally independent of the request of your national funding body. So for the finance particularly, you have to follow their rules. It's very important. In PRIMA, we just want to have an idea of how much you have spent in your project. We don't want the details of each item, etc. You will see. On the platform, you will have the Excel file, the budget file you have submitted with your proposal. And so this will be your original budget, and you will just have to report how much you have spent. Not very detailed, it's just a global idea. After, you have to report to your funding agency according to their rules, their own rules. We are not intervening in that at PRIMA. The final reporting, I will not repeat, it will be exactly the same way of working. Of course, the technical report will be a little bit more extended because you have more results, more things, maybe more impact, we hope. And so this is it. For the financial report, I just leave this information because in PRIMA we need certificate of financial statement, etc. This is for section 1, but it's to let you know that even for your funding agency, sometimes you have to give certified account. So have this in mind. This is just for information, it's not a mandatory things. Financial aspect, so I repeat, follow the national regulation of your country. PRIMA has no action on your budget or other things. What we can do, it's to make recommendation to you. It's very important to verify before doing any expense on your project that these expenses will be reimbursed. They are eligible. So the only person to know that is your NCP, your national contact point. I put here in red something very, very important. It's to keep record and support document the originals for 5 years. It's very important to be well organized since the beginning, to have all your invoice, to have the contract, to have anything related to the project, because in PRIMA we have the obligation to audit 1/3 of the beneficiary. So the probability of being auditing is high. And when the come, you know, you want to see the document very clear, very organized, to avoid any problem. So for this reason, I strongly recommend you to keep all your documents very well organized and safe. Because for 5 years, because even if your project is has a duration of 3 years, the audit can be done until 2 years after the end of the project. So for this reason, 5 years is necessary. And the audit can be carried out by somebody of the foundation, but it can be carried out by the European Union or the OLAF or another subcontractor. Now, about the dissemination of your results, the open access, the visibility of the PRIMA funding. So it's important in such project to have an active dissemination and communication activities. You want the people to know what you are doing, and in PRIMA we push to have an impact. So to be close to the smallholders and to promote what you are doing in your research. So it's very important. This has to be reported in the midterm report, of course, but we really encourage you to have a lot of communication and dissemination activities to ensure a strong impact of your project. Intellectual property right, of course, if you have patent, if you develop new varieties, etc., etc., all this has to be very well explained in your consortium agreement. Who will be the owner of the patent, how it will work, if you have a license, etc., etc. Here again, PRIMA is not intervening directly, but this has to be very clear in your consortium agreement. Because at the beginning of the project, everybody wants to share the data, but sometimes after, at the end, some institutions are more reluctant to that. So it has to be clear since the beginning who will be the owner and what are the rules that will apply for that. And of course, if you have patent and these kind of things, in the monitoring platform, you will be able to fulfill this data and inform us about that. Now, about the publication and the open access, as we are part of the H2020 program, we have the obligation to put the data on open access. So you have different options to do that. The goal of open access, of course, sometimes it's costly. You have to see with your funding agency how these costs are eligible or not, or how you can manage that. But sometimes also I attract your attention to the green open access that is much cheaper and that can be a very good compromise, and that is fully accepted by PRIMA. For your communication, so in all your communication, you have to use the EU and the PRIMA logo as you are part of this program. PRIMA is funded at 50% by the EU program, so both logo have to be together on all your presentation. After also, I believe that the logo of your national funding agency can also be requested. This depends on what is written in the contract you have signed with your national funding agency. In general, you have to put also their logo. So the logo of all the partners that are participating to the given work. If it's in the papers, in a scientific paper, for example, in the acknowledgement section, you have to mention that you have received a financial support by PRIMA. That is a program supported by the European Union, and also to put your national grant number. So I believe this is it. I thank you very much for your attention, and I'm more than happy if you have questions to answer you. Thank you very much.
Thank you, Fabrice, for this very interesting presentation regarding the general rules for project monitoring, especially for HaloFarm for HaloFarm project. I don't know if we have questions. We have 5 minutes if you want to ask a question. If you have question, you can raise your hand. I see Christian. Please, Christian, you can ask your question, please.
Yes, thank you, Fabrice. I wonder whether it was possible to get these slides. in order
Sure.
to manage.
This is what I'm doing. I'm uploading it in the chat. I hope you will be able to download it.
Okay, thank you.
And if not, I will send it to Karim, and he will be able to
Yes,
diffuse.
yeah.
Yeah.
Thank you.
Thank you.
Any other question, please? Dr. Wasif or Dr. Hassan Shah, you can ask your question, please. Just activate your.
Okay. Thank you very much, Fabrice. We appreciate your good presentation, actually. And I support our colleague question concerning having this presentation with us to follow what is the regulation of PRIMA and how can we do with the data online and this and that. So we would appreciate if you forward this presentation to us. Okay?
Sure. Yes, the presentation is already so in the chat of the meeting, and I will send it to Karim also if you want to send it to all of you.
Thank you very much. Thank you.
Yes, I acknowledge the receipt of the this presentation by Fabrice in the chat. You can download it from the chat. Any questions? Otherwise, we gonna move to Hervé, please.
Sorry. Thank you very much for your presentation. In regard of audit, do we have to for some institute? This is sometimes internal. So I understand that we have to keep all the documents for 5 years. And should we include the fundings for audit in some institute, or is it going to be paid basically by HaloFarm and by PRIMA? Sorry, is it clear?
Yes, you mean if the cost of the audit is covered by PRIMA or by your project?
Yeah.
Yeah. No, normally it will be PRIMA. If PRIMA or the EU is sending an auditor, it's on their own cost. It's not for the project.
Okay, thanks. Thanks very much.
Dr. Ahmed Youssef from the Desert Research Center.
Yes.
You can ask a question, please.
Okay, thank you, Fabrice, for this clear presentation. My question is, how about the password accessibility to enter the platform of PRIMA?
Yes, in reality, so all the coordinator of the 2018 project have received this file. You are part of the 2019, so you will be in the second wave, but I mean in less than in 15 days, I think more or less, the coordinator will receive this file, and we will be able to give you access. So because before we have to create the account for each partner, each person. So once this will be done, we will inform the coordinator, and then so you will be able to access this platform.
Thank you very much.
You're welcome.
Fabrice, I have when I check the chat, there is our colleague Antonella Castagna from Pisa, from University of Pisa. She is asking if the presentation requires an access key.
This is strange. This means you cannot download it like this?
No, we can't. I can't download it either. So.
Okay. So the best, I will send it directly to you, Karim, and you can forward to your partner. I think it's the best option.
Okay. Thank you. Thank you, Fabrice. So we will gonna move now to the main part of this kickoff meeting. It is related to the HaloFarm project, and I will give the floor to Ahmed to share this session. Please, Ahmed, you have the floor in order to introduce this session.
So now, thank you, Karim. So we will, as Karim said, so we will move to the project presentation, and we will start with the coordinator, namely so Karim, who will present the project in terms of objectives, approach, and impacts. And then we will move to the presentation of the different project partners involved. So from all our the countries, partner countries, and then to another part which is about dealing with the work plan, the work package, deliverables. So let's start first of all with Karim. So the floor is yours.
Thank you, Ahmed. Just I will share my presentation. Okay, thank you very much again for making the space and for taking the time to join. First of all, I would like to thank and congratulate all the participants and partners in this HaloFarm project for their inspiring and fruitful collaboration, and also for the huge work and the excellent work they did from the beginning. Just to remind you that HaloFarm is a project, a PRIMA project that is selected in response to the call 2019. It is relevant to the thematic area farming system and section 2 topic use and management of biodiversity as a major level of sustainability in farming system. The project is a RIA, is a research and innovation action. The total requested budget is about 850,000 euro. The duration of the project is 36 months, beginning from 1 October or the 1st October 2020. This project, as I already mentioned, is coordinated by the Center of Biotechnology of Borj-Cédria from Tunisia, in collaboration with the Tunisian Center of Water Research and Technologies, also in the Technopark of Borj-Cédria. We have also the Center of Marine Science for Portugal, the Spanish Research Council from Spain, the University of Pisa from Italy, the University of Occidental Brittany, UBO from France, and also the National Institution of Research for Agriculture, Food and Environment, INRAE, also from France, and also we have the Desert Research Center from Egypt. This project is centrally organized by PRIMA and funded by national funding agencies from participating states. From Tunis, we have the Tunisian Ministry of Higher Education and Scientific Research, and we have the INPR, the National Agency of Promotion of Research, which manage the funding. We have of Scientific Research and Technology. From France, we have the ANR, l'Agence Nationale de la Recherche. From Spain, we have the State Agency of Investigation. From Italy, we have the MUR, the Ministry of University and Research. And from Portugal, we have the FCT, the Foundation for Science and Technologies. Let's remind you the specific challenge and scope of the topic of this project according to the PRIMA Annual Work Plan 2019. The major threat to the Mediterranean area and also to the planet is the agro-biodiversity depletion, because these agro-biodiversity depletion reduce variety of available food, and also it reduces the ability of agro-system to respond to environmental challenges. We have also another challenge, which is related to the monoculture approach of intensive agriculture, to climate change, and the reduction of soil fertility that decrease significantly the ability to naturally cope with the biotic and abiotic stresses for maintaining high agricultural production. What PRIMA are asking from the proposal? PRIMA asks that the proposal shall implement activities contributing to the development of biodiversity-based agriculture in the Mediterranean countries. Proposal should also analyze how traditional agricultural systems make use of biodiversity. And also, PRIMA are asking for integrative approaches that exploit synergies and minimize problems due to monoculture. They are also searching and looking for complementarities between scientific and stakeholders' approaches and knowledge. And to address these challenges and to be in line with the scope of this topic, HaloFarm will contribute significantly to the optimization of innovative, eco-friendly, and sustainable agricultural and production system in salt-affected Mediterranean soils using the desalination and economic potential of halophyte. Why halophyte? Halophytes that are those plants that we can find growing naturally in saline biotopes. These halophytes have many and have already many ecological uses and economic uses. Example of ecological uses: halophyte can be used for soil and water desalination. They are used in phytoremediation, landscaping, carbon sequestration, and also dune stabilization. They have already economic uses, such as source of food, bioactive molecules, forages, and animal feeds. They have also ethno-veterinary uses, and they are also sources of biofuels. In HaloFarms, we will mix the cultivation of halophyte with conventional crops on salt-affected soils through two farming practices: crop rotation and intercropping. The biomass that will be produced by this farming system will be valorized for food, pharmaceutical, and veterinary application. Also, the biomass, and to ensure sufficient and safe biomass from halophyte, we will optimize in vitro cultivation protocols that will be also useful to preserve endangered species and to overproduce bioactive substances. HaloFarm also will include environmental studies to address the environmental performance of using halophyte for soil desalination. It will also include socioeconomic studies to assess the benefit of the new farming system and product on the engaged smallholder farmers. And third, it will include a market uptake study to exploit the added value product from halophyte. All these studies are performed in close relationship and partnership and collaboration with the local farmers, the stakeholders, and social actors. We will set up and we will optimize and implement 4 experimental pilot sites in this project. We will have a site in Tunisia, in Spain, in Italy, and also in Egypt. Now, what are the expected impact from HaloFarms? HaloFarms, at the end, should reduce salinity in salt-affected agricultural Mediterranean soils. We expect also a better growth, productivity, and quality of conventional crops when halophytes are planted prior or in combination with these crops. Shift from monoculture to polyculture also is expected to restore biodiversity. The rapid clonal propagation and conservation of germplasm of halophytic species by using new in vitro methods for their multiplication. We also expect to find new added values product and application from halophyte biomass and extract. We expect that more local small farmers and stakeholders will be engaged in these new farming practices. New market opportunities of halophyte product as source of food, pharmaceutical, and veterinary will be also optimized. We also expect to enhance the capacity building in halophyte farming and production system. And more interestingly, we expect, as PRIMA also asked, a reinforced partnership between researchers, farmers, and stakeholders. Thank you very much for your attention.
Thank you. Thank you very much, Karim, for this overview and for this general presentation, which enables us to better understand the project objectives, approach, and the expected results. And I think it's in well conjunction with this, we will continue with the presentation of the project partners now. And I will start with Professor Hamza Al-Fil from the Centre de Recherche et Technologie des Eaux, so CERTE, in Tunisia. So, hello, Professor Hamza.
You heard me?
Yes.
Yes.
Would you please? Yes, it's fine. Okay. So the floor is yours, please.
Okay, I will share the screen. I have 5 slides, and I will not take a lot of time, maximum 5 minutes. Okay, hello everybody, and I thanks all of our partners in the project, this project HaloFarm, and especially Professor Karim for all his effort. Donc, I am Hamza Al-Fil. I am professor in chemical engineering and specialized in the desalination field, especially reverse osmosis membrane and reverse osmosis. I am also head of the laboratory desalination and natural water valorization, which belongs to the Center for Water Research and Technology, CERTE, which is in the same technopark as biotechnology centers. The CERTE, our center, contains 5 laboratories and 3 specialized units. The different laboratories are georesource laboratory, desalination and natural water valorization, and the 3rd laboratory is for used water and environment. The 4th laboratory is water membrane and environmental biotechnology, and the last one is the laboratory of water discharge treatment and valorization. The societal challenges of our centers are the water source mobilizations, water quality improvement, local water management, and eco-innovations. This is the principal themes of our centers. For our laboratory desalination and natural water valorization, the discipline of the laboratory is water sciences, and the principal research themes are improvement of drinking waters, membrane desalination of brackish and seawaters, valorization and reduction of desalination brines. And this is by this field we will help in this project, HaloFarm project, because we know one of the problems of desalination systems is the brine, which have had high concentrations, and when we are far from the sea, usually we find problems for the rejection of the brines, and now we work for reduce and also valorization of the brines, and we will test the possibility of the valorization of brines for irrigation of the halophyte. The next step are the prevention against scaling, and finally improvement and management of water irrigations. The principal activity in our laboratory in relation with HaloFarms are, the most of them are related with desalination, as the project of in collaboration, German-Tunisian bilateral collaboration, which called the desalination nutrient recovery and diversified valorization technique of nature-based recycled wastewater. We have another project with society, international society, which have two great farms in the north of Tunisia and the south of Tunisia. And this project called brine valorization and optimization of performance of water desalination unit for irrigations. We have another convention with the partner which will be a partner of us in this project, and they have a farm here 5 kilometers far from Borj-Cédria Technopark, and he have citrus and also a desalination unit, and our collaboration was called the diagnosis of well water desalination unit producing water for citrus irrigations. We have also another project with German Arab Transformation Partnership, Salinity and Transformation. Also, we worked on valorization of brackish-water aerobrine for tomato irrigations, and we have annulled FP6 projects, which call it open gains, and the project is interested to water desalination by the use of different energy renewable resources. That's the essential information that I will share with you. And I thank you for your attentions. Thank you, Mr. Ahmed, Professor Ahmed.
Thank you very much, Professor Hamza Al-Fil, for this nice presentation. Our second speaker will be Professor Hassan Shah from the Desert Research Center in Egypt. So we will share the presentation actually that you already have.
اتكلم الموبايل ازاي ناخده ازاي ونور لنا النور عشان نشوف من هنا. لا انا اتكلم من هنا ولا اتكلم من
Can you already see the presentation?
Okay. Good afternoon, everybody. Do you hear me?
Yes, of course.
And can you see me now?
Yes. Yes.
Okay, good. Thank you very much. First of all, I'd like to thank Karim and my close friend, Dr. Shadli Abdelli, and Dr. Ahmed Al-Baz, and all my colleagues in Borj-Cédria, and our new colleagues from different countries in the project. You are very welcome, and we are happy to join you in this fruitful project. First of all, let me introduce myself. I'm Dr. Hassan Al-Shair, the ex-Vice President of Desert Research Center and the Director of the Egyptian Center of Excellency for Saline Agriculture. This very specific center was established in 2013 to work directly with the problem of saline, whether saline soil or salinity in water irrigation. And this center actually has been involved in the government strategies for reclamation and the cultivation of the huge project in Egypt. Huge agriculture project. It is a mega project in Egypt for cultivation of around 3 million acres around. This center, we call it in brief ECISA, or Egyptian Center of Excellence for Soil and Agriculture, has been working on distance and as well as with the research activities, of course. So we are working parallel in research and the agricultural development on most of the desert area in Egypt. And this center actually also has been involved in conducting series of projects, different agriculture aspects in the northeastern and in New Valley, some desert area of Egypt. And the center has around 50 experts representing all the disciplines of agricultural development. We have a soil scientist headed by Professor Muhammad Wasif, and the groundwater research headed by Professor Ahmed Yusuf, who are with us now. And we have our colleagues from plant and from, vegetable scientists, crop, field crops, and horticultures, and livestock, socioeconomic people. So we are representing different aspects really in this center. And we have been engaged in bilateral project with Borj-Cédria, with Professor Shadli Abdelli and my friend Karim for over 4 years now. So we are glad to join another project, particularly with new colleagues from Italy, from Spain, from France. So everybody are very welcome to join you. Okay. Concerning that, we have already conducted several projects related to HaloFarm, and since we are interested in halophytes and salt-term plants for a long time ago, I think we started together with Dr. Shadli in 1995 with Professor Helmut Lief from Germany and some colleagues from Italy, Portugal, and Spain, and France as well. So this project HaloFarm is a continuation of our efforts, and I'd like to just throw some information about the contribution of the project in Egypt. The farmer groups will be the Desert Research Station, the Matruh Research Development Center, and the extension services will be Matruh Research Development Center and Agriculture Department, and in Matruh Governorate. Matruh Governorate is located in the northwestern coast of Egypt, adjacent directly to the Mediterranean Sea, nearby the Egyptian-Libyan border. The participating in this optimization or implementation of system will be Al-Mogra, Alliance for Agricultural Development. And as I said, Al-Mogra is one of the huge mega agricultural projects in Egypt nowadays. And the dissemination channels for this project is Matruh Research Development Center, and the advisors will be from Agriculture Department and in Matruh Governorate. The contribution of Desert Research Center or Egyptian Center of Excellence for Saline Agriculture actually will contribute in HaloFarm project through several work packages. The first work package is the work package 2, is maximizing mixing halophytes and the crop species and salt-affected soil, where the leader is the P7 and the participants in PI, P2, P3, P4. And also we will contribute in work package 4 for the valorization of high added value products, where the leader is the PI and the participants is P3, P5, P6, P7. Egypt will have the leadership for the work package 5, which is entitled the Environmental and Economic and the Applicability Assessment. And of course will also contribute in work package 6. Concerning, I will talk for to describe the contribution, the Egyptian role for work package 2, mixing halophytes and crop species in salt-affected soil. And we are starting. Can you move to the second slide, please, Ahmed? Okay, work package 2. Yes, mixing halophytes and crop species in salt-affected soil. Not this one, Ahmed. The one before. The one Dr. Ahmed Yusuf sent it to you. Now, with the work plan. Okay, so now this is the last slide, Ahmed. I'm talking about the second slide. Okay, never mind. So the contribution of DRC for the work package 2. We are starting the cultivation of salicornia and the tomato this week. Our delay was due to the following reason, actually. The receiving of the National Security Agreement, which was done this week, only this week, waiting for the transfer of the first advanced payment from the funding agency. This is the main two reasons for our delay. But thanks God, we got already the security, and we are dealing with our national agency, and we're starting already to work on some propagation of salicornia and the tomato to be ready to be transferred at the farm. I see the project area. However, we'll start to do the following activities. Number 1, to collect and analyze the soil samples from the selected field. Number 2, prepare the selected field for cultivation. carrying out the irrigation network at the farm to test the water samples of the ground water and to cultivate, of course, the seedlings in the selected fields after the propagation. The last activities will be implement of the proposed planning design, and of course we will stick with the formal design for all the group as indicated in the slides for cultivation and intercropping system or mixing halophyte with the crop species. I think this is all for the now still some activities here. Collect and analyze the soil samples from the selected fields, prepare the selected fields for cultivation, irrigation network implementation, we mentioned that, and testing the water irrigation from the groundwater, setup of the halophyte intercropping system in the selected area, data set the changes in the salinity and the chemical properties of the soil during halophyte cultivation, okay, and data set on mineral content of crop plants and halophytes under intercropping, data set on physiological and biochemical parameters of crop plants and monoculture in intercropping with the different halophytes, data set on yield and the quality of fruit produced in monoculture and intercropping. It takes the month 18th to 30. Predictive statistical model correlating tomato growth, productivity, and the quality with the soil and environmental parameters. Biomass of desalting species and of crop species to deliver to the work package for their characterization. And this is all for this, I think, for this session. If you have any question or any comments, I'm ready to answer. Thank you, Dr. Ahmed Baz.
Okay. For this first presentation. And now, so we move to Professor José Antonio Hernandez from the Spanish National Research Council, CSIC.
Hello. I'm
Hello,
going to upload my presentation.
Hello, Professor José.
Oui. Okay. It's okay. Can you see the presentation? Okay. Hello. Well, as you say, my name is José Antonio Hernandez. I work in the CSIC. At present, I'm head of the Department of Plant Breeding in my institute. Okay. Okay. Well, my institution is the Spanish National Research Council, or the CSIC in Spanish. It's an honor institution founded in 1939, but in fact, this institution is the successor of a previous institution called Committee for Extension of Studies and Scientific Research, created at the beginning of the 19th century, and whose first president was Professor Santiago Ramón y Cajal, that was Nobel Prize just the year before. Okay, the research at present, the research in CSIC is structured in three main areas: society, life, and material, which cover more than all the human knowledge. And we are about 1,500 research groups and about 120 institutes in all the country. Okay, my institute is CEBAS, is founded in 1944. It's one of the most important institutes of agricultural science from the CSIC. And we are structured in 6 departments and 11 research groups. We are about 67, probably 70 senior researchers and about 250 staff members. Okay, the singularity of our institute is in fact, we are a multidisciplinary center with 3 different areas of research, because we research from soil, in fact, natural resource, agricultural science, and food science technology. We research from soil to the consumer, passing from the plants. Here at the bottom is the webpage of our institute and the blog that we manage. Okay, we take part of the group of Free Tree Biotechnology, and in the staff of the research include me as main research, Dr. Abel Piqueras, that is an expertise in biotechnology, and Dr. Pedro Díaz Vivancos is an expertise in plant physiology and biochemistry. But also we are other collaborating researchers, Dr. Gregorio Barba, that at present he has a contract partially funded by the European Union. So for this reason, he cannot take part at the beginning in this project. But at the end of this contract, we can contract Gregorio with this project. Dr. José Ramón Acosta Motos is working with us. We are going to contract him very soon with a company project, but also he collaborated with the Catholic University of Murcia. And Mrs. Verónica Becerra is a student that, a postgraduate student that now is working with us, in fact, in this project as practice student. Okay, we have a lot of facilities in the institute, some service like metabolomic service, genomic, in vitro culture, and growth chamber. We have also an experimental field of 32 hectares and 3 laboratories in the group. Okay, we are involved in work package 2, 3, 5, and 6. You know, work package 2 is about mixed cultivation of halophyte and cross species in salt-affected soil. We are going to study the growth performance and physiological and biochemical trait of commercial tomato plant by analyzing different parameters. The work package 3, in fact, we are the leader of this work package 3 about optimization of in vitro cultivation of halophyte. We are going to set up protocol for in vitro micropropagation of different halophyte plants. And also, we are going to characterize the acclimatization to ex vitro condition of the propagated plants. We are also involved in work package 5 and in work package 6 about the dissemination. We are going to contribute actively in this part. We hope to publish some paper, to send some communication to Congress, to use the blog to inform about the project. Also the social network, the press news, and also the contact with farmer and agricultural cooperative. In fact, we have a contact with a farmer association that they are very interested in this project. They are collaborating with us, and they are organizing a meeting, and they are going to use some of this project to communicate to other farmers. Okay. In relation to work package 2, we are going to contribute with different tools for the evaluation of the physiological and biochemical state of commercial tomato plants grown in the presence or in the absence of halophyte plants. We are going to perform first soil analysis, the mineral nutrition, just to study the ion homeostasis in the plants, the antioxidant metabolism. We are going to study the chlorophyll and flavonoid contents and calculate the nitrogen balance index. Also, we are going to study the chlorophyll fluorescence parameter, and we are also study the production and the quality of the fruits by using different parameters like bleach degree, acidity, or sugar content. This is some of the of the device we are going to use. Okay, relating the work package 3, also we are going to contribute with different tools for the evaluation of the physiological state of the micropropagated plants during the acclimatization to sweeter condition, in this case of the halophyte plant. We are going to use the chlorophyll fluorescent evolution just to analyze how the plants are starting to make the photosynthesis process. Also, we can use the different oxidative stress parameter. This is the case of lipid peroxidation. Just we can see the evolution from in vitro to the in vitro condition after 2 days. We see an oxidative stress, but then the plant is acclimatized to the new condition. Or also, we are going to study the evolution of different antioxidant enzymes. Just to study this process. This result corresponds to the acclimatization to stevia plant that we published previously in a special issue in antioxidant and was selected for the cover for the special issue. So we hope to do the same with the acclimatization of the different halophyte plant that we are going to investigate, and we hope to publish this result in open access journal. So thank you for your attention.
Thank you very much, Professor José Hernandez.
Okay, thank you. If there is any question?
I think we keep questions for later, please, because we're a little bit running out of time, and it's I think it's better to finish this part at least, and then we will see.
Sorry, I don't understand. Can you repeat the question?
I told you, since you are running a little bit out of time, so let's move to the other presentations, and then we will see if there are some questions. Okay, thank you. So we continue now with Professor Christian Magnier from the Université de Bretagne Occidentale, UBO in France. Hello, Professor Magnier. Professor Magnier.
Hello.
Hello.
You can hear me? Yes?
Yeah.
Okay.
Yeah. The floor is yours, please.
I share my screen. Can you see my my slides?
Yes.
Is it okay?
Yeah.
You can see it?
Yes, like this.
Okay. So, hello everybody. I'm Christian Magnier. I'm working at the University of Brest in France. And this is a multidisciplinary university with all fields represented: mathematics, physics, biology, social and human science, and so on. University of Brest is located over 3 different sites in Brittany, which are about 70 kilometers far from each other. On the whole, the University of Brest hosts 1,350 individuals in about 34 research units. Most of these individuals are scientists. And it also hosts 20,000 students, and its budget represents approximately 185 million euros per year. As for its scientific production, University of Brest produced more than 1,000 papers and 5 patents each year. And an important point is that collaborations with startups generally result in the creation of joint startup, in average 8 each 5 years. So in the University of Brest, my research unit is called Geoarchitecture. So it's also a multidisciplinary unit hosted by the Faculty of Science. This unit includes 27 individuals. 24 of them are scientists, and also we have this year 14 PhD students and 4 postdocs. Annual budget of my research unit represents more than 5 million euros each year, and scientific production represents more or less 40 to 50 papers each year. So also biologists, scientists of the unit works in collaboration with the industrial fields, and we create or we accompanied creation of startup. More or less one small enterprise is created each 5 years. So this we are interested. As for the biological part of my unit, we are interested in halophyte physiology, biochemistry, and biotechnology. So we are working on halophyte responses to salt stress, to UV stress mainly, and we try to isolate metabolites important in these plants and biological activities of interest for food or cosmetic applications. Therefore, to achieve this work, we have collaborated with some of HaloFarMs partners for a long time. You can see here 3 representative examples of these collaborations, mainly with CBBC, the Tunisian partner. We are collaborating since 2008, 7 or 8. With 2 PhD theses in co-supervision, 2 postdoctoral projects, and as a result, we have published about 16 papers and deposited 1 patent. Also, a more recent collaboration with the Italian partner of HaloFarMs and the Portuguese partner of HaloFarMs also involve 1 thesis or postdoc work and several papers already published or in preparation. So within my research unit, HaloFarMs project involves 3 scientists, including me, 1 technician and 1 PhD student. Our contribution to the HaloFarm project mainly is found in work package 3, which is called in vitro cultivation of halophytes for salt remediation and agriculture in saline soil. So mainly we are going to collect natural halophytes in their natural habitats. And then we are going to sterilize some parts of these plants or seeds and initiate in vitro cultivation of these plants. These in vitro halophytes will be evaluated as for their ability to uptake salt from the medium, and by comparison with several species of halophytes, we will be able to select the more efficient species, and within species, the more efficient clones as for their ability to uptake salt. Then we will use these selected species or clones to optimize the micropropagation on salt medium. Then these efficient plants will be acclimatized to glasshouse culture, or then to open field culture, and then will be provided to partners of Work Package 2 to their crop rotation or intercropping work. Also, we will have a small contribution on Work Package 4 as for characterization of main compounds and biological activities in these plants, mainly for food application and maybe also for health applications. So thank you for your attention.
Thank you very much, Professor Manier. We have another participant from France, namely now Professor Hervé Host. So, Professor Host, you ask that we share your presentation. Is this right?
Yeah, can you hear me and see me? Can you hear me?
Yes, we can hear you, yeah.
Okay, and maybe see me also. So I'm very happy to join you and to join this kickoff meeting, and thank you for the organization by your Tunisian colleagues. So I don't know if you can see my presentation or not.
Yeah, it will come, I guess. Well, not yet.
Sorry for the delay.
No problem.
So
we have a copy actually. I don't know. Would you like that we share it?
If you have the copy to save time, yes, please go on. It's okay. So to save time. So my name is Hervé Host. I'm working as a director de research senior director in INRAE in a mixed unit of research in Toulouse in combination with the vet school, and so I'm a vet by. And I'm also working for this institute, which is considered as the main research objective research institute in Europe in regard of agriculture and environment. And in regard of the project, we close my camera. In regard of the project, I'm partner 6. Do you see my slides?
No, not yet, actually. Oh. Okay.
So to describe my work, I'm working in vet parasitology, so basically worms, which are a main parasitism of sheep and goats when they are bred outdoors. And these parasites are producing production losses, but they can also affect animal welfare and provoke some clinical size and sometimes mortalities. And according to FAO, thank you very much. I can see my slides, so I hope everybody can see the slides. So according to FAO, second slide, please. Sorry, Karim, and thank you for your help. These parasites are already considered as one main pathogens for both animal welfare but also human welfare. And one of the main way, because of this importance, economical and also health importance of these animals, of these worms, sorry, one of the main way to control these parasites have been based on the use of chemical drugs described as anthelmintic drugs. And can you see the slide? I don't know if you see the slide. So, do you see the slide?
Yes.
Yeah. Okay. So if you are
on the second slide.
I don't see them, so it's not so easy. Sorry for this. So if you are on slide 3 with a very sad sheep with the cycle of the nematodes, so we were using chemical anthelmintics to treat the worms, but now we are facing several limits. One is increasing societal demands to reduce the use of chemical drugs in agriculture and in farm breeding. Also, an increasing constraint on regulation of chemical use of treatments, and also resistance to anthelmintics in worm population, which is a worldwide phenomenon. So, if this is slide number 4 with the future of control of these worms, parasitic worms, we are now exploring and moving towards an integrated sustainable management of gastrointestinal nematodes. And one of the options in red, I hope you can see it,
yes,
is the use of plants and natural compounds, which is basically back to some traditional use of natural products to control these worms, these parasitic worms. So it can have impact on the production of small ruminants, both sheep and goats, and but also can serve as a model for human nematodes. And moving to slide 5, thank you, Karim. In regard of the participation of my team to this HaloFarm PRIMA project, as you can see on the top of the slide, we will be involved mainly on WP4, which is in regard of valorization of the product biomass and possibly extract of these halophyte plants. And within this WP4, which will be led by Louisa from CCMAR, we will be mainly involved in the assessment of possible use in vet science in regard of parasitic infection of sheep and goats, with some in vitro assays and in vivo anthelmintic activities, and also check the possible effect on meat and milk quality. This is within a collaboration for nearly 5 years now with and also Marta from the same group, and with collaboration also with Pisa University in Italy. And thank you for your attention, and sorry for the technical troubles. Thank you very much to Karim.
So thanks a lot, Professor Host. I mean, we were able to follow you. I mean, both with regard to the sound, but also you will be able to follow your presentation.
Thank you.
Now, thank you. Now I move to Professor Anna Maria Ranieri from the University of Pisa, UNIPI, Italy. So, hello, Professor Ranieri.
Yes. Just a moment. I try to share my slides.
Otherwise, we can share it.
Okay. If not, we can share it actually. Yeah? Yes, this is quite challenging.
Can you see?
Not yet.
Do you see my slide?
No, not yet. Elle est encore connectée, elle n'est pas connectée. Professeur Ranieri.
Elle a déconnecté, Karim. Elle va connecter, je crois.
It looks like Professor Ranieri was disconnected. Can come. So we'll wait just a little bit if she is back. Otherwise, we will move to Professor Luisa Custodio. So wait for her for some seconds.
Do you want me to try to share my slides?
Yes.
Okay.
Yes, please.
Let me see if it works.
Sorry for this disconnection, but problem connection. So I can begin again or after Luisa?
I'm just trying to share it.
Okay, so I wait for Luisa and I speak after. Okay.
Okay. Can you see my presentation, Karim?
Yes.
Okay.
Are seeing your screen.
Yes. Yeah. Okay. So it's very nice to meet you, although only online. I hope that the COVID will go away and we can meet personally shortly.
Excuse me, Lisa, maybe I cannot see your presentation. Maybe nobody can, or it's only me.
And now?
Just open the
No, I cannot see
presentation,
anything.
Luisa, open the presentation.
Yes, but it's open. Cancel it and do it again.
Sorry, Luisa, I don't see it too.
Would you like that we share the presentations from here?
Yes, let me just try one more time. Maybe I can do it. Okay.
In the first step, we can open the presentation and share your screen.
And now you can see it?
No.
So something, it's not working.
So we can open it from here.
Yes, please.
Okay.
Thank you.
Okay.
CCMAR.
Can everybody see the presentation, please?
Yes, now I can see it. Thank you.
Yes.
And the other colleagues? The
Yes.
other colleagues? Okay.
Yes,
Yeah.
now I can see.
In France, it's okay.
Everybody can see it?
Yes.
Yes.
Okay.
Yeah, okay for Hervé.
Okay, thank you, Karim. So you can go for the next slide, please. Okay, so I am Luisa Custodio. I work in CCMAR. Oh, now it disappeared, Karim.
Because
Now even me, I cannot see it.
Did you touch something or?
No, nothing. It was on the first slide and then it disappeared.
And for the others?
Yes, this is
I can see.
Slide disappeared,
Hervé.
yes.
we only see with Luisa.
Yes. Well, I can talk, but it's boring if you don't see the slides. It's strange. What had happened here?
It's always a pleasure to see you, Luisa.
Thank you, Hervé. Let me try to share it again, and if it doesn't work, I will just talk what I was supposed to talk. Because now it doesn't open the PowerPoint. Karim, can you try to share it again, please, from your side?
We are doing this actually, but there are many screens that are open. I don't know.
J'ai partagé.
Okay, now I can see it. Okay, you can pass to the next. Okay, it's fine for everybody.
Yes.
Yes.
Okay, so I will try to be brief. I am Luisa Custodio. I work in CCMAR in the University of the Algarve. I have a graduation in agronomy and a PhD in plant biotechnology. And in the past 10 years, my specialization is the biotechnological valorization of natural products from marine organisms like algae, for example, but also halophytes. I am the group leader of the Extreme Bio research team, and currently I have 4 projects, all funded by the Foundation for Science and Technology, and all of them are related with PRIMA. My research group has different research areas. The first is extreme foods, in which we valorize the natural products from different species, especially in the food industry. The next area is extreme pharma, where we do the same thing, but targeting the pharmaceutical applications, like, for example, isolated bioactive compounds and food supplements. We also have extreme pharma, where I work in extreme vet, where I work with close collaboration with Hervé, where we try to identify natural products, especially from halophytes, can have veterinary applications. Since we have already identified several halophyte species which are very interesting, we are now developing the extreme crops that aims to do a sustainable cultivation of selected species in greenhouse. And we also have the extreme micro, where we try to micro-propagate and apply other in vitro techniques for the propagation of selected halophyte species that allowed very good results in assays, also to achieve the production of target bioactive compounds. As I told you before, I work in CCMAR. CCMAR is a leading international marine science research center. In CCMAR, we have different groups working in different areas, ranging from ecology to aquaculture and to biotechnological exploitation of marine organisms. And we have access to Rio Formosa, which is a very, very interesting and rich ecosystem that provides access to a plethora of highly interesting species. In these projects, our responsibilities and roles will be to do the biochemical analysis of biomass produced by the partners in different methods of, in different cultivation methods. This biochemical analysis includes, for example, the establishment of the nutritional profile and also to see if they have components that display some bioactivities that are interesting for us, like antioxidant, for example, anti-diabetic, neuroprotective, and so on. We will also collaborate in the oversee and assist in the biochemical analysis performed by other partners, for example, in the veterinary area. And we will also collaborate with the project coordinator, Dr. Karim, and other partners in the management and dissemination of the results of the project. You can pass for the other slide, please. What are our expectations from the project? I have a lot of expectations from the project, and maybe the most important is to increase knowledge of the use of these halophyte species in different cultivation systems, saline cultivation systems, that can be translated and applied to different areas in Portugal, because here we have a lot of soils that are becoming saline and then cannot be used for the traditional cultivation of glycophytes. So these forms and these consociations would be very interesting to be applied here. We also want to increase scientific knowledge by collaborating with different partners from different countries with different and complementary expertise, and also to boost the sustainable use of different halophyte species, not only as agronomic cultures, but also as sources of high-headed commercial products. And that's all for now. Thank you very much. If you have any questions, just tell me.
Thank you very much, Professor Custodio. Well, thank you very much, Professor Custodio, for this presentation. And the last one will be from Professor Anna Maria Ranieri. So if you want, we can share this presentation.
Okay, I'm trying.
Okay, we let you try first.
I'm trying. Oh.
On n'arrive pas. Elle n'arrive pas. Elle n'a jamais comme UNIPI.
Do you see my presentation?
No.
Oh, yoy.
We can try to share it from here, Professor Anna Maria.
Okay, okay.
Présentation. Is it okay like this?
Yes.
Yeah, you can.
Just a moment.
Yes.
Because I don't see. Just a moment.
You
Okay.
don't need to share by yourself. It is already shared.
I see. Okay, good morning. Finally, I am Anna Maria Ranieri, the principal investigator of the team of the University of Pisa, an Italian public university founded in 1343. The second slide, please. Okay. Researcher participating in HaloFarm project, work at the Department of Agriculture, Food and Environment that include experts in many scientific disciplines related to agriculture, food action and quality, plant genetics, biochemistry, biochemistry of plants, primary and secondary metabolism of plants, physiology, and environmental management. My department has many laboratories, experimental field, and greenhouses for protected cultivation, and provided the scientific and administrative support for the execution of the specific tasks of HaloFarm in which it is involved. Together with the Department of Agriculture and Food Environment is involved another department of Computer Science, with expertise in data management, bioinformatics, and artificial intelligence. Both departments have many young people in training, PhD students or postdoc recipient, and temporary research. No, the second, I need the first one. Okay. And both departments develop research topics with a high degree of national and international cooperation, and annually host numerous visiting scientists and scholars from many European and non-European countries. The second slide, please. Okay, the researchers involved in HaloFarms of the Department of University of Pisa, beside me, are Antonella Castagna, which is expert in plant biochemistry and food quality, Luca Incroci, Professor Luca Incroci, with expertise in horticulture and crop modeling, Marcello Mele and Giuseppe Conte, both experts in animal feeding and sustainable livestock systems. The researchers from the computer science are Stefano Chessa and Paolo Milazzo, with expertise in predictive models and application for smartphones. The other slide, please. Okay. Within the project, University of Pisa will lead work package 2, mixing halophytes and crop species in SAL-affected soils. And it will be detailed in the second session of the meeting, and will be involved in the following work pages. As all partners, UNIPI is involved in work package 1, project management, and in training, dissemination, communication, such as work page 6. And in addition, UNIPI is involved in work page 4, valorization of the produced biomass for mixing cropping systems, and in work page 5, environmental sustainability, economic and applicability assessment. UNIPI expect to acquire advanced knowledges on the capability. No, I don't see the slide. Oh, okay. So UNIPI expect to acquire advanced knowledge. Okay, it's okay. On the capability, on the capacity of the halophyte plants in intercropping or crop rotation to remediate saline soil and ameliorate crop productivity and quality, to improve its expertise on the possible commercial exploitation of edible halophytes, and to increase knowledge on the use of halophytes in the formulation of the diet for farm animals, but also for human people. And development of an app for smartphone to assist the farmers in the decision of whether and how growing the main crop in mixture with halophytes species. Thank you very much. This is the first part of my presentation.
Thank you.
Thank you very much, Professor Ranieri, for this general presentation. With Karim, we'd like to suggest that we take a 5-minute break, if you do not mind, and then we will come back with the second part of this kickoff meeting. Is it okay?
Yeah, just a question. I think Christian asked the question about the availability of the slideshow. Can you just indicate how it will be possible? Because I think clearly some of my slides were not obvious for everybody. So, but as a general thing.
With Karim, so Karim told me that for the second part he has already all the presentations, so you already sent them to him. So, and if you do not mind, we will also continue by this way. So we will share the presentations from here.
Yeah, but if you wish to get access after the discussion and after this presentation, how can we get access? That's my point.
I can send you all the presentation, of course.
Yes, thank you, Karim.
Okay. So it's now about 3:20. So let's. I suggest that we meet once again in 5 minutes. Let's say to 25 or 26 past 3.
What do you think? It's okay?
It's okay for me.
Yeah. Just to prepare the second session, okay? That is related to the work package plan, work package presentations.
Perfect.
Okay. Hello, everybody.
Hello.
Hello. So we will start now the second part, dealing with the work plan, work packages, and the deliverables.
كده شايفني يا كريم؟
Yes.
كريم.
So actually, it was thought that Karim will start with the project management first.
Karim.
Le son n'est pas clair.
Karim.
So the first.
Le son n'est pas clair.
Yes, yes.
Karim. Are you ready, Karim?
Please deactivate your mic.
Oh, c'est flou, le son c'est flou. C'est Ahmed, on ne vous entend pas bien.
Il y a beaucoup d'écho. Il y a beaucoup d'écho.
Oui.
Karim. Ahmed.
Je ne sais pas vraiment, ça me surprend.
Ahmed. The voice is not
Naam.
clear. The voice is not clear. The voice is not clear.
I hear you. I'm here. I heard you.
You hear me now?
Yes, yes, yes.
Okay, good, good. Okay.
And you?
Yeah, I can hear you now.
Okay. Okay. So let's move forward now with the second part. Please deactivate your camera.
Professor Hassan, please deactivate your camera, Professor Hassan.
Is it okay now?
And the mic too, please.
Is it okay now?
No, it's not okay.
Not yet?
Not yet.
Is it okay now?
Yes, but the mic also should be deactivated, please. So we move now to the second part. So initially it was thought that Karim will start, but we thought that it would be better to let the Tunisian presentations as the last ones. So when we start with Professor Ranieri once again about the work package 2, about mixing halophytes and crop species in salt-affected soils. Are you ready, Professor Ranieri?
But I think it is better that Karim could share the slides.
We will share the slides. We will share the slides.
Yes, yes. Yes, thank you.
Is it okay like this for everybody?
No, I don't see anything. Okay.
And now?
I see. Okay. It's okay for me. And for the other people, it's okay?
Yes, it's okay.
Okay. Work page 2 is led by University of Pisa. No, sorry. Okay. And it involves no, the second line, the second slide, please. Okay, it's okay. Dunque, work page 2 involve partner 1 at the CBBC location, and partner 2 at the CERTE location, partner 3 at the DRC location, and partner 4 at the CSIC location. Okay. Worker period 2 will start at the month 1 and end at the month 30. The second slide, please. Okay. The main objective of work package 2 are to assess the capacity of intercropping with halophyte to remediate saline soils. The second objective is to evaluate the ability of halophyte to reduce salt stress in crop plants and to increase crop productivity. And the objective number 3 is to collect edible biomass and bioproduct for biochemical and analysis foreseen in work package 4. The next slide, please. The work package 2 is divided into 3 tasks. The first one deals with the setup of mixed cropping trials in 4 countries: Spain, Tunisia, Egypt, and Italy. At least one same halophyte and crop combination will be used by all partners involved in this task, and the trials will be evaluated following the same protocols. Tomato was chosen as the standard crop species, and the salicorn as the salting species. 3 types of plots will be used. 1 combining tomato and halophytes, 1 containing only the halophytes, and the third one containing only tomato. The second year, tomato in monoculture will be grown in the halophyte plot to compare the desalting capacity of the halophyte monoculture with that of the mixed cropping. Task 2 foreseen a series of analysis of soil and plant, as well as the assessment of tomato productivity and quality to monitor the capability of the selected halophytes to extract salts in the soil in the first 30 centimeters of soils, and to monitor the capacity of the system to decrease salt stress, so to alleviate salt stress in the glycophyte plants. Moreover, the correlation of tomato growth, productivity, and quality with the soil and environmental characteristics and parameters will be investigated through a predictive statistical model that will allow to estimate crop performance and tomato productivity also under conditions differing from the experimental ones. The third task consists in collecting the edible biomass of crop species such as tomato tomato fruit at the red ripe stage and of halophytes, as well as the product. All samples will be dried and or lyophilized and delivered to the partners for biovalorization on work page 4. The next slide, please. Okay. Regarding deliverables, the work page 2 deliverables are listed in this slide and specifically consist in setting up 4 target sites in 4 participating countries to conduct field intercropping experiment using halophytes and tomato under site-specific saline conditions. The second is a measure of soil pH and electrical conductivity, total organic carbon, total nitrogen, or anions and cations at the beginning and at the end. It is possible to see my slide or not?
Yes.
For some people it is not evident the slide on the okay. So for that for the second deliverable, it will be carried out the measure of soil pH, the electrical conductivity, total organic carbon, total nitrogen, anions and the cations at the beginning and at the end of cultivation, mono and intercropping. The third is to measure anion and cations in tomato and in the intercropping halophytes. The fourth is a data set on photosynthetic performance, carotenoid, chlorophylls, and phenolic levels, and antioxidant defense mechanism of cultivated plants. Data set on productivity and commercial quality of tomato fruit cultivated in monoculture or mixing cropping. The other one is a predictive statistical model correlating tomato growth, productivity, and quality with the soil and environmental parameters. Samples of edible parts of commercial species and halophytes and of bioproduct for work page 4 analysis. And samples of specimen of halophyte for their natural, their edible part and byproduct for delivery to work page 4. I don't know if I can continue. If it is possible, I speak also about the last slide, the next steps for my work page is to set up the experiment trials in spring 2021, measurement of soil parameters before and after plant cultivation, measurement of mineral content in plant tissues, both in crops and in halophyte plants, collection of biomass from cultivated halophytes and crop and from halophytes in their natural habitats and delivery to work package for partners. Okay. Thanks for the slides, Karim. Thanks for the slides.
Thank you very much, Professor Ranieri. Was everybody able to see the presentation, please?
Yes.
Okay. So we will move to work package 3 from our Spanish partner. So Professor Gregorio will talk about this.
Yes, I'm going to try to upload myself the presentation. I'm doing it right now. I hope it works because before in the test it was working. Let's see. Yes, it's uploading. Takes a little while.
Okay, it looks good.
Can you see it?
It's been uploaded.
Okay.
Yes, it's fine. It's fine.
Okay. Yes. Okay.
So please go ahead. Thank you.
Yes. Okay. Good afternoon, everybody. It's a pleasure to meet you be here introducing the work package 3. This work package we are the leaders here at Csic of this work package, and the work also will be conducted at the CBBC and at the Université de Bretagne Occidentale. Firstly, I thought it was worth mentioning some recent taxonomical changes that has been reported for the Amaranthaceae family. We have found that Arthrognemum macrostachym has changed genus to Arthrocalum macrostachym, with mainly an inland distribution here in Spain. And also due to some recent molecular studies, the presence of Salicornia fruticosa has been discarded in Spain. Instead, 2 Salicornia species are found, Salicornia hispanica, with an inland distribution, and Salicornia lagasci, mainly found in coastal sour marshes. Other genera to be used during the consumption will be Chenopodium, Sesubium, and Cridmum. And the picture shows an example of coastal landscape where Salicornia lagasci is predominant vegetation here in Murcia, our region. Okay, in synthesis, the deliverables for this work package can be summarized as follows. A sampling of halophytes from their natural habitats, different plants, optimization of the in vitro propagation, which will comprise sterilization methods, selection of plant material, growing conditions. I can read that someone cannot see my slides, but I hope it's only one person because I cannot do anything. Okay. And then also screening in different media and evaluation of superior lines in which the response of the plant material to growing and soil concentration will be key. Then acclimatization to ex vitro condition of these elite germ plants which respond better to the soil treatments, and characterization of the plant performance and physiological stress parameters during acclimatization. In other words, we can also say that the main goals to be achieved are evaluation of unexploited germplasms, which will contribute to maintaining these species from their natural habitats. By using this, sorry, I didn't want to do that. Yeah. By using this in vitro platform, we could avoid seasonal variation found in the field and would provide clones free of diseases for the rest of participants in the consortium for their use in greenhouses experiments, even for the nurseries. And it also would be of great importance to establish a comparison on the plant performance and desalting capacity between the acclimated plants and the commercial specimens already available. And now a very brief slide about our current and very recently initiated experiments here at Csic. During last month, we conducted 2 samplings and an inland location where the vegetation is dominated by Arthrochilum macrostachym, and soil samples were also taken associated to the sampling spots. And we found that Arthrochilum macrostachym displayed 2 plant developmental stages: bigger plants at seed production and younger and shorter plants. We also took seeds from some specimens for long-term storage and especially for in vitro germination experiments. And very briefly regarding the soil analysis, we found high sodium and chloride content, which is expected. We also found very high calcium concentration. That is due to that the soil is calcareous, so that should be the reason. And we also found extraordinarily high iron concentration, and this can be due to contamination of the soil from the surrounding industries. We are waiting for having the results of the nutrient analysis in the plant tissue to see if there is any comparison or correlation between the levels found in the soil and the levels found in the different plant tissues, the leaves and the roots. Regarding the initiation of the in vitro culture using explants, the sterilization was followed by cultivation on MS media with 15 grams per liter of salt plus growth regulators. We distinguish between younger and older plants and between apical and basal nodal segments. We also tried the effect of different fungicides, and we also add during the sterilization a mix with ascorbate and citric acid to avoid oxidation. As a result, we found that apical segments of older plants respond better to sterilization so far. We indeed have a very high or quite high contamination rate, probably due to the high abundance of endophytic species, at least in the location and in the samples we have conducted. So we have a low survival rate. However, among the survival explants, we are having a relatively high sprogion rate, as it can see in some of these pictures. In parallel, we are conducting in vitro germination experiments, where after sterilization, seeds are put on MS medium with or without salt. We found out that germination works better in the absence of salt, whereas seedling growth is much better in the presence of salt. In fact, you can see here in the upper picture how the seedlings grow in the absence of salt. They are not doing well. They have some symptoms, stress symptoms probably, and they have this red discoloration, whereas the one in the other pictures looks much better. So, and also it's interesting to point out that we did not find contamination. So that is a good thing. And so the initiation of the experiment from seeds looks promising so far. And the last slide, just to say thank you to everybody, and that we hope that in the coming months to have advanced in the development of this work package, and of course have incorporated at least the Salicornia species found in Spain to the in vitro platform. Thank you very much for your attention.
Thank you very much, Professor Gregorio, for this nice presentation.
Thank you.
It's a pleasure to see that there are already ongoing activities that started some time ago. And
Yes,
it's really nice to see that you have already preliminary results.
Yes, thank you.
Okay, thank you very much. We move to work package 4, which deals with the valorization of halophyte biomass. So once again, it will be a pleasure to see Professor Luisa Custodio, if I'm right.
Yes, thank you, Karim. Do you want me to try to make the presentation, or can you do it by yourself? Maybe it's easier.
We will share your presentation, Luisa.
Okay, thank you.
The same one, like the same one, CCMAR.
Is it okay?
I still cannot see it.
Just a moment, please.
Okay. Can you go, please, where I ended in the first presentation, please?
This one.
Okay, yes. Okay, so my role here is to coordinate work package 4, which deals with the valorization of the produced biomass, aiming its potential use in different areas, like, for example, food, pharmaceutical, and the veterinary. Our main goals will be to gain knowledge on the chemical composition of produced biomass, either the edible parts or the non-edible parts, or maybe we can call it the parts with less commercial value. To do that, we will do the analysis of the nutritional profile of the edible parts, including also the presence of anti-nutritional components. Then we will prepare food-grade extracts, namely ethanol, acetone, and water from the less valued parts of the produced halophytes, and evaluate those extracts for chemical composition, or including also the nutritional profile. Then we will determine in vitro and in vivo bioactive properties which are relevant to the use of these extracts in food, pharmaceutical, and veterinary areas. And finally, we will assess the bioaccessibility of key nutrients and bioactivities in edible parts and extracts through an in vitro model of human digestion to see effectively if the extract contains one bioactive compound, if after ingestion by humans, it will still be available to be absorbed by the human body. Can you pass, please, Karim, to the other slide? This work package has 3 tasks. The first one is comprises the valorization of the biomass for human nutrition and for human health improvement. In this task, we will have to establish the nutritional profile, the presence of anti-nutritional elements and also toxic factors, and also to make evaluation of the microbiological contamination of that biomass. And then we will perform the extraction of natural products and evaluate the toxicological and microbiological safety. And finally, on the task 413, we will evaluate the nutritional and bioactive properties of the extracts, focusing only on those which are not toxic and which don't have toxic levels of microbiological contaminations. In task 4.2, we will then assess the biomass aiming its veterinary uses. For that, we will establish the nutritional profile, in vitro digestibility, and fermentability of the biomass. It will also make the study of the in vitro anti-helmintic activity of selected extracts, and also the in vivo anti-helmintic activity of the active extracts, and also assess the effect of the administration of those extracts on the health status and on the quality of the animal products, namely meat and milk. Finally, on task 4, we will make we will select the extracts that were more active and more promising in all of these biological activities that may effectively have commercial interest, and then we will make a complete metabolomic characterization of those extracts. Can you please pass it on, Karim, please? We have different tasks and deliverables. The other one, please. Okay, I will not go through, but the main deliverables is effectively to be able to indicate the biomass which is able for human consumption, to have extracts which are non-toxic and microbiological safe, to be able to identify extracts that present adequate nutritional profile and with in vitro functional properties, to be able to identify the extracts with in vivo functional properties, to be able to identify halophytes with veterinary applications, either as biomass or as extracts, and finally to make a standardization of the extracts, that is to be able to identify marker compounds that are present in relevant extracts and to go forward to commercial applications. Can you go to the next slide, please? Because in this task, we are going to work with biomass produced by the other partners. We are still not working on those biomass, but I think we are able to receive some biomass, for example, from Italy. So the next steps will be to receive that biomass from the different partners and process it, and then to establish the biochemical profile of that biomass and start preparing the extracts that will be delivered for the different partners to be evaluated for in vitro assays in the first step, and then select the bioactive, then that will proceed for in vivo assays. And I think that's all. Yes, it's all. Thank you.
Okay. Okay, thank you very much, Professor Custodio, for this detailed presentation, but focused but detailed presentation. Now it's time for work package 5, which is entitled to Environmental Sustainability, Economic and Applicability Assessment, which is presented by the DRC.
Okay.
Please, Dr. Karim, can you share the presentation from you?
Yes, it will come.
Okay, Karim, I'm ready. Put the bed document. Okay. What's the problem, Karim?
Professor Hassan, are you ready?
Yes, I'm ready.
So the presentation is already there.
I cannot see it. Where is the presentation? Karim? كريم انت قفلت التليفون. اوكي اوكي can I start كريم؟ can I start كريم؟ شغلوا كده التاني التليفون مش اللي عامل الكلام.
Yes, Dr. Hassan, you can start your presentation, please.
Okay, so I'm talking about the contribution of DRC for B3 as a B3 in working package for 5. Okay, working package 5. The participants are B1, B4, B7, and B8. The purpose of this activity is to establish the exploitation of the research results through one, assessment of the environmental performance and economic relevance of optimized farming and products. The second is the establishment of processes to translate them to business. The third is the capacity building and the dissemination of the project output among stakeholders such as farmers, industrials, end users, and policy makers. I think the participants will integrate their results obtained in working package 2, 3, 4 to draw general conclusions and future recommendations for their effective implementation and wide applicability. In addition, to determine to what extent optimized technologies contribute to filling knowledge gaps at the Mediterranean scale. The main output of Working Package 5 includes several activities such as environmental studies to address the environmental performance of using halophytes for saline soil remediation. The second is socioeconomic studies to assess the resulting socioeconomic benefits of the new farming practices and the products on the engaged farmers. The third output is an economic evaluation through cost-benefit analysis for each system to educational, in addition to the educational impact of extension activities such as methods and results demonstration, field visits, farming school farmers, and extension publication, such this kind of materials. Concerning the socioeconomic activities, it will include cost and returns, which will be estimated for each method or agricultural system in order to conduct an economic feasibility study and an economic, social, and environmental evaluation. It will be done through the payback period, added value, net present value, benefits and cost ratio, internal rate of return, sensitivity analysis. So I think all the participants will calculate and get the values of these parameters. Extension activities will contain farmer field school, field visits, training, and making workshop for farmers selected. Extension publication, which will be translated result to farmers in forms of pamphlet or brochures or booklet or any kind of extension publication. which will be translated the results to farmers and other stakeholders. The different investment costs, all costs of establishing the project, for example, the value of land, equipment, drilling wells, and various devices. The fixed cost will be calculated, whatever is rent or taxes, variable costs, whatever seeds, seedlings, cost of raising irrigation water, machine maintenance, all these fixed costs will be calculated from each side. I mean from all the participants. We expect that from this project, from this activity, or from this work package, that the expected outcomes are reinforcement of DRC expertise on the use of intercropping system to remediate saline soils and increase the productivity of commercial crop and salt affected soil. This is the general outcomes of the project for DRC actually, not only for the package 5, but the contribution of DRC in general. The second expected outcomes will be introducing an integrated model for better utilization of salt affected resources. Number 3, introduce new salt tolerant crops for multi-purposes usages in the region. Number 5, increasing farmers' skills and developing and adopting crops that improve soil properties and increase its production. Create new job opportunities and increase the participation of gender, women and men, and increasing their skillfuls. Adopting new crops on which to base an industry and increase farm animals, reclaiming the land and making sustainable development in the region. Improve the quality of life in the selected area, increase capacity building, increase farmers' income and the standards of living. I think some of the participated countries in this in work package 5 will expect the same expected results and outcomes. And we will be glad to be the header of this work package and to cooperate with the participants, colleagues from different countries. And I think that's all for us. Thank you very much. That's it.
Thank you very much, Professor Shahab, for this presentation. It was clear. We continue now with 2 presentations from Karim, dealing with the project work package 1, project management, and and work package 6, training, dissemination, and communication. And we will start by the last one, so work package 6. Okay, please.
Okay. It's all right.
Is it fine for everybody?
Yes, it's fine.
Okay.
Yes, okay. Work package 6, training, dissemination, communication. This work package is very important for our project. The leader will be the Center of Biotechnology Borj-Cédria, and all partners are involved in this work package. The objective of this work package is to acknowledge the project outcomes to a wide range and diversified targeted audience, including the general public, academia, trainees, industry, investment, and finance actors, social and environmental policy makers, researchers, farmers and their association, technical and employed members of various stakeholders, as well as educational institutions. This work package aims to train and exchange students and also disseminate and communicate results. We have different in this work package. We have dealing with the training and students cross-discipline integrated in the project, like, for example, soil desalination, like in vitro cultivation and multiplication of halophyte, valorization of halophyte. We will also train PhD students, MSc graduate and non-graduate students. So this project can be very and an excellent opportunities for students to be well trained and to perform excellent research. The task 2 is about the dissemination. We plan to participate to international conferences, and we think that we can participate to at least 3 international conferences per year. We will organize 4 seminars in participant countries that match the short-term mission of the coordinator. We will organize also 3 international workshops, and also we will be able to publish scientific publication, at least 15 in international peer-reviewed journal, open access. For the communication, we have also planned many communication activities. For example, logo, and we have already succeeded to prepare our logo for this meeting, and you can see it. It is halophyte. You can see the blue color. It represents the Mediterranean area. It also represents the salinity, and you can see the green color, which refers to the environmental and let's say the environmental and the ecological benefit of our project in farming system. We also plan to have a website. It is under construction. It will be hosted by the website of our center, the Center of Biotechnology of Borj-Cédria, and I see that some partners have already included our project in their website, like, for example, the University of Pisa, like the CSIC, the Spanish Council, also INRAE from France, and so on. And I hope that the other will do the same. We will be connected also to social media networks. We have already created a Facebook page, and you can see the address. And I invite you and all the participants to this kickoff meeting. I invite them to visits this page, you can find all the detail of our project, and in the future you can see all our activities and achievement. Instagram, Twitter, and LinkedIn account also planned to be created. For the promotion material, we need to prepare flyers and roll-up that can be used when we will assist to workshops, to international conferences, and so on. We also will also have press release at least once a year. We scheduled presentation in schools, universities, and other organizations, and also we will organize as a communication tool open days. It means visits, guided visits of general public, trainees, stakeholders, investors, etc., to the experimental fields that will be implemented in the four sites, and also we will also visit to one that is organized by the Ministry of Higher Education and Scientific Research with all these partners, and also in collaboration with the European Union. It will be presented, and you can find all, let's say, the information about our participation on 27th of November 2020. Now I will move to my presentation regarding the project management. Like work package 6, the project management will be also led by the Center of Biotechnology Borj-Cédria, and also all participants will be involved, of course. The objective of these very also interesting and important work package is to carry out an efficient project management that maximize the responsibilities and prevent accumulation at the coordinator level. We also scheduled different tasks, beginning from the strategic and technical project management, and these tasks it will be managed by a management and coordinating structure. This is the management structure of HaloFarms. You can see that we have a scientific committee, and also we have a general committee, and we will have also other boards and panels that will assist the scientific committee and general assembly in the coordination and management of the project. We will have an advisory board, we will have an innovation and exploitation panel, and we will have also an administrative and financial panel. Of course, all decisions will be made by the scientific committee in collaboration and in consultation with the General Assembly, and we will have also close interaction with the PRIMA Secretariat and also with the national funding agency from the participating state. We have another task besides the first one. We have a task dealing with the risk management in order to monitor deliverable failure and mitigation measures. For this task, we should present to our let's say organization, to the funding agency, and to PRIMA a quality and risk management plan. This table shows you what are the risks that can indicate our activities. And in the proposal, we listed some risks. For example, we mentioned risks that are related to cropping studies. For example, unforeseen climate events, insects, diseases, and in the right, you can see the proposed mitigation measure. So, for example, for this risk, we think that it is interesting to strictly monitor the experimental trial and to use corrective measurement promptly, immediately. For example, we have risks related to the valorization of biomass. We have risks related to management. For example, we can have some delays in respect to the established timetable, and we think that we can tackle this risk by communicating well and also early enough. I also drafted, this is not mentioned in the proposal, but I also drafted some risks that are related to now the actual crisis, pandemic, the COVID-19. So we can think that we can have some limited capacity due to home officing, to home schooling, to sickness, to quarantine, and in this case, we can think to apply for a project extension, to have some delays of deliverables, etc. Also, a very major risk that is related to COVID-19 is the limited mobilities. And it can happen, it will happen, and it is happening for some of our students. Mobilities and training by students and by researchers will be limited because there is introduction of to go abroad. There is closing of borders. So we can tackle and handle this risk by organizing, for example, e-learning session, by organizing webinars like our webinar of today. The last task related to project is the data management. This is also very important task that should be also considered for our project. The data that will be generated for our project will be shared in the open research data pilot in Horizon 2020, following the principle as open as possible, as closed as necessary. The data generated will be findable, accessible, interoperable, and reusable. The data will be also collated and stored in a database hosted by the Center of Biotechnology of Borj-Cédria data server. Of course, we will deliver a data management plan, I think, after 3 or 6 months from the beginning of our starting date. The result related to the new farming system will be disseminated without restriction. And the data related to the new product and application will be protected through the appropriate intellectual property mechanisms. Just to mention that we have already prepared and signed a consortium agreement by all partners in accordance with PRIMA rules to clarify potential IPR matters. And I thank you very much for following the details of work package 6 dealing with dissemination and communication, and work package 1 dealing with project management. Please, Ahmed, the floor is yours.
Thank you very much, Karim. Well, I guess you can hear us, at least all of you. Okay. So thank you very much, Karim. Thank you very much to all our speakers and partners involved in this project. Despite some technical challenges, I think this also belongs to this project. I mean, we have to adapt, and we will learn how to manage such sometimes some technical difficulties. But I think overall, everybody could listen and also see the presentations and could be informed about the content. I personally enjoyed attending this meeting. It was really useful and focused for me. I mean, I learned much more. Well, I had some discussions with Karim before this meeting, but now I have a better idea about the project in its conception, objectives, philosophy, and also in terms of what we expect from this. I really enjoyed to see how teams involved in this project are complementary with respect to their expertise, and how they are also multidisciplinary. This is also, I think, an interesting dimension, even I would say also a very important dimension, since we have different aspects, not only the scientific, but also, I mean, the sociological, but also the human aspect is also quite important in this project. And I think all of these points are strength. And I do hope that we will be able to succeed in attaining our objectives. And I'm really confident about this. I had the opportunity to see that all of our partners, they have strong expertise once again, and they have, I mean, they are also very well equipped, and there's also we are complementary. So I wish you all the success in this project, and it was also encouraging for me to see that there are already some ongoing activities which started some time ago. This is the case, for instance, in Tunisia also. And I'm sure that we will have the opportunity to share data and to see concretely progress made and achievements, hopefully. I don't know, when do you expect to have another meeting? I mean, even webinar, but I think while waiting for improvement of the situation related to COVID, we might think about organizing regularly such webinars. I think this would be also a good opportunity to follow the progress made. And from our side, we will support you, Karim, and all of you. We will provide full support in our lab and our center. And I wish you personally much success. So thank you.
Thank you, Ahmed. I thank you very much for sharing this session and for all these concluding remarks dealing and that present all what has been presented by our partners. I would like to invite you, if you want to interact, to have some comments. If you want, I don't know, you can raise your hand. I will give the hand to Fabrice. Please, Fabrice, you can.
Yes.
You have the floor.
Thank you very much, Karim. Thank you very much to all the presenter of this afternoon. It was very interesting for me. It allowed me to have a much better understanding of what you are doing precisely, and each one, what is the work that will be carried on. So your project is quite big. It's very nice because, as you said, there is a complementary skills and this is very appreciated. I really like the part that you will deal with biomass, but also bioactive compounds, extract salt from salty soil. So this is wonderful. As in all the project, there is a lot of work to do. There is a lot of deliverables. There is a lot of things to do, to realize, to carry on. And so for this reason, I want to remind you that this is a collaborative project, and of course, each of you has to collaborate as much as possible by being on time, by facilitating the work of the coordinator, because as coordinator, you have to gather all the information for the consortium. So it's not an easy task. And of course, it depends also on the commitment of all of you. So this is very, very important. I appreciate also the part you at the end when you talk about the contingency plan. So to have a plan B in case something is failing in the proposal, this is very, very important. So very good to remind this part. I just have a small question, or I'm just worried about one point, is how you will involve the stakeholder really in your project, because I've seen, so you have a part in the lab, you have field trials, you will have dissemination, but as you may know, sometimes dissemination is not enough, because if you don't have the stakeholder since the beginning and they want to try and to test maybe your methodology, your new varieties, this is my question. How do you think you will be able to involve really the stakeholder and so to valorize what you are doing in your project? Thank you very much. But overall, it's a very good project. Congratulations.
Yes, thank you, Fabrice, for your remarks and also for your encouragement. I will just jump to your question. The stakeholders are parts of our project. We are sorry because we did not present the potential stakeholders, and we have many stakeholders that we listed in our proposal, and I think we can organize a seminar or a webinar to involve mainly the stakeholders that will be involved in our project. For example, we have local farmers that are engaged in the different countries, with the different partners where the pilot sites will be set up. We have also dissemination channels. For example, we have agencies that will disseminate our results. For example, the INPR, for example, the National Agency of the Promotion of the Research, will help us to valorize and to transfer our results to the stakeholders. We have also partnership with association, with NGOs, with non-governmental associations that are also engaged and that are already well identified by our project and also by all the partners. So the stakeholders will be at the heart of our project. They will be involved in all the steps of our project, and they will be informed about all the achievement and all our activities. It should be, of course, done locally. Each partner will talk and will meet the stakeholder in each countries, and of course we will have the general committee besides the scientific committee, and the General Assembly is composed of coordinator, partners, and all the participants in this project, including stakeholders. So stakeholders will help us and will be our force, our power to and will assist our decision, and also will help us to make our project and our results attainable and also successful.
Thank you very much for this reassuring answer, because yes, as you said, so you have many stakeholders, but maybe I miss, or it was not visible in the presentation of this afternoon, that you have NGO, you have association. So this is very important, because as you know, sometimes the research projects are doing a lot of work, but in the end it stays in the lab and it never go outside. So it's very important to be in touch with these people and to also develop the project together, not to just have a top-down approach, but a constructive approach. So I'm very happy with your answer and reassured. Thank you.
Thank you very much. Thank you. And I think it is very important to organize as soon as possible, I think, a workshop that gather the researchers and the partners involved and the stakeholders. Why not?
Yes, sure. I believe it's very important to have them every time you can, to be sure they are really following and you are interesting them, and so they will uptake and upscale your project in the field.
Yeah, thank you.
Thank you.
If one of the partners can also interact with this aspect, or you can, Professor Hassan.
Salicornia. As you know, we have local Salicornia is Salicornia rubia, and this strain is very slow and gross. So my question is, we will have to use Salicornia biglavi, for example, or we can have some seeds from our colleagues from Spain or from Italy or from where. So the clear question is, are we allowed to use Salicornia biglavi or not? This is my question.
I don't know, Professor Anna Maria or Antonella, can you please, if you have answer to this question? Otherwise, I see that I can say that we have chosen Salicornia with other species, with Suida and Atropimum, but we don't specify the species. So you can use Salicornia bigelovi. You can use also Salicornia arabica, Salicornia europea. We have, each partner can use the species that is suitable for his activities. Please, any other comment, please, or conclusions before we close? Dr. Ahmed.
Thank you, Karim, for this organizing, very good organizing and all presentation. My questions is, in the meeting, Sky meeting on 28 May, the Pisa participant illustrated his cultivation through greenhouse. It is possible to cultivate in greenhouses or in open field, either of them?
Yes, I think that it is better to test and try in open fields.
Yes.
This is my answer. For example, in Tunisia, we will work in the open fields with local farmers here.
Okay, Karim, thank you very much.
Any other, please, if you have some conclusion about the kickoff meeting, so we will have other opportunity to discuss our activities. In terms, yes.
Gary?
Yeah.
This is Hervé.
Pepe, Hervé, si Pepe, si Hervé. Allez-y.
Sorry. Well, I think one of the challenge could be to improve the technical issue between us through the internet system. And because Fabrice is here, could we ask to improve it and ask whether through the HaloFarm and the PRIMA project, is it possible to ask for some fundings to help us in regard of getting access to these general professional Zoom or general team?
Yes, if I may answer, I believe this is up to you and in agreement with your funding agency. If the coordinator can buy a license, for example, a professional license for Team or Zoom or whatever, first you have to check that is eligible for your funding agency. This is the only thing I can say. But many times in reality, it's the institution that has institutional access to this kind of platform. Sometimes it's WebEx, sometimes it's other thing. So I believe maybe that you have to investigate a little bit what is available in your institution first, and then to see whether it's suitable to buy a more professional system. But if your funding agency, if this cost is eligible for the funding agency, PRIMA has no problem with that, sure.
So this is eligible?
No, you have to check with your funding agency. For PRIMA, there is no problem. But if your funding agency tell you you cannot buy a communication system with the grant, you have to find another way.
Okay, and could it be eligible for, we were speaking about end user organization. Can we expect or suppose that to diffuse the result of HaloFarms to make a link with a group of end users or farmers?
Sorry, I'm not sure to understand very well your question. Do you mean like you want help to promote your project?
Yes, in order to promote the project. This is related to the WP6 dissemination and implementation. Can we help farmers? And I think one of the WP5 is also related to smart diffusion. Can we use some fundings from the sorry, from HaloFarms to implement that? And how can we use it? And how can we implement it?
But in reality, when you have submitted your project, you elaborated a budget. And so in this budget, in front of each item, you put an amount. I mean, if it's for communication dissemination, you should have a part of your budget that is reserved for this. And if this has been approved by the funding agency, I mean, there's no problem to spend it. But is that the question?
No, to make it clear, is it also possible to include Zoom inclusion of Zoom connection and teamwork connection.
Yeah.
Professional.
Yeah. Yes, as I told you before, in reality, the rules of fundings are really the national regulation. So to be sure, I don't know the 19 regulation of all the countries that are participating, because this is a very precise point. Can I buy a communication system with the grant? If you were in section 1 with the fund of the EU, I will tell you, yes, sure, no problem. But after, as you are funded by different countries with different rules, you have you are in France, so you have to call the ANR and ask them if it's eligible or not. And if you have planned in your initial budget something like this?
I think it was scheduled. I don't think it was directly scheduled, but I can include it as consumable, I suspect. But I'm asking also a question for everybody, every team.
Yes, but the answer is the same in reality. It's about asking your funding agency if this is eligible or not. And of course, there is something very important. Each partner does not have to buy a license. Maybe only the coordinator can buy it for the group, for the project, not each one of you.
I'm not speaking about myself, about organization and possibly about farmer organization. And is it eligible, since one of the objectives is dissemination towards end users? Can we have to attract end users, and can we to attract them, can we include a possibility to have a connection towards Zoom, or at least they will not pay?
Yeah, but I mean, if you are the owner of the license, for example, if I have the license for Microsoft Teams, I can invite anybody I want. They don't need to have the license to join. I just send the link and they click and they attend the meeting. So you don't need to buy a license for them.
Yes, but I still maintain my comment.
Okay.
It's good to attract. It's good to attract the farmers.
Yes. Yeah.
To give them something.
Yeah. What Hervé said is very important, and also the response of Fabrice. Also, I think it is correct, because all funding will be managed by the national funding agencies. So we have to discuss and to talk with our national funding agency if it is possible to have this kind of funding and this cost. And also it depends of the cost of each partner. So for example, some partners, they don't have sufficient budget to for example to fund such materials or such connection. Thank you very much, Hervé, for your comment. Can any other comments, any other questions, any other concluding comments? If there is no questions and comments, we come to the end of this kickoff meeting. We hope that you have now a clear idea about HaloFarms, a general clear idea, and also about the partners that are involved. Of course, there are some insufficiency to be tracked, to be corrected, to be handled for the future, mainly to connect with this new virtual tools of communication, and we will do. We hope that we will be able to be successful in achieving all the objectives and all what we scheduled as activities. I would like to thank and congratulate again all the partners in HaloFarms, and also to thank all the participants to this kickoff meeting for interacting. I would like also to thank Fabrice from PRIMA. PRIMA is our support, is supporting foundation. I also don't forget the National Agency of Promotion of Scientific Research, and especially Professor Shadli Abdelli, the General Director, and my colleague Sonia Zgarni, the computer responsible here for facilitation of this kickoff meeting. I thank you all, and I hope that I can see you and meet you again as soon as possible and in other webinars. Thank you very much.
Thank you.
Thank you, Karim.
Thank you, Karim, for your work.
Thank you very much.
Thank you, Karim.
Amazing.
Karim, may I have
I will-
one